All right, thanks very much, Cheryl, for the kind of introduction. Just incidentally, I was boosted 3 weeks ago, and I got a negative COVID test yesterday, so for anyone in the room. Phil's a tough act to follow. So Phil has shown you a highly proven and incredibly transformative technology, one of the most transformative that we've seen in medicine over the past decades. Today, I'm going to talk to you about an entirely unproven technology that will emerge over hopefully the coming decade or two to also transform medicine. So as Phil already motivated, there's a pressing need for new gene therapeutics, in particular in the delivery space. So we have, of course, gene modulatory compounds, but we don't have the ability to deliver them specifically to organs, tissues, and cellular subtypes, as well as for vaccines. We know there are now over 6,000 genetic disorders. Cancer has claimed over 600,000 lives in 2019 in the US alone. COVID has claimed 5 million lives worldwide. Of course, we have vaccines, but we would also like more effective ones that have longer lasting immunity. And we should not forget, of course, another pandemic that has been scourging the planet for decades now, which is AIDS caused by HIV-1 that has claimed over 33 million lives worldwide. So thinking about delivering gene therapeutics, I'm going to just highlight sort of two technologies, and some pros and cons. So LNPs, lipid nanoparticles, that was the subject of the previous talk, have now, of course, impacted medicine in a real way in patients with a number of approved drugs that are clinically licensed also. They have a low cost of production relatively. They have versatile control over the gene therapeutic payload, including siRNAs, ASOs, mRNAs, and so forth. But they also have pretty proven now limited targeting capabilities. In contrast, if you think about AAVs, which R&D has pumped billions of dollars and continues to into the development of, these viral vectors have incredibly efficient targeting and transgene delivery to specific organs as well as cell subtypes. They have an exorbitantly high cost of production that we haven't yet been able to bring down, and they're also limited in their control over the gene therapeutic payload to a 4kb transgene. What I'm showing you on the bottom is the particles that our lab is exploring. These are virus-like particles that sit in between viruses and lipid nanoparticles because we have, on the one hand, full control also over the therapeutic modality, which can be incorporated siRNAs, ASOs, mRNAs, as well as CRISPR-RNPs, as published recently by Jennifer Doudna's lab, as well as peptides and proteins, and also targeting modalities such as sugars, peptides, that can be presented on the outside of these particles, much like in a virus, in a multivalent way. But these are also chemical compounds. We make them synthetically. The cost of production is somewhere in between LNPs and viruses. That, of course, will depend on how production is scaled up. And unlike these two other modalities of delivery, they're completely unexplored in their capabilities for organ, tissue, and cell subtype targeting. In our lab, we're exploring a few different angles with these virus-like particles. On the one hand, we formulate them out of a messenger RNA. So we take a messenger RNA and we actually fold it up into a virus-like particle using complementary oligos. We also encapsulate CRISPR-RNPs within these particles. And the subject of today's talk will be exploring how we can use these particles to mimic viruses in their structure and function by targeting subsets of B cells for vaccine applications. Some of the properties I already went through on the right. The size of these particles is on the order of 10 to 100 nanometers, which we control fully together with the structure and composition. Another important point is, of course, they're made from nucleic acids themselves, so they have immune cell stimulation, which I'll show you some early results on, which we are able to control in some ways. And importantly, these is thinking about clearance mechanisms and ultimately, eventually, FDA approval, hopefully. On the one hand, these are, of course, more complex entities. They're not just a single compound with a nucleic acid like a GalNAc. These are more highly structured and consist of other components. but they also biodegrade in vivo, which can be an attractive opportunity if one is actually able to control these for the right application. So the technology is rooted in a 2006 paper by Paul Rothermund. People might have heard the term DNA origami. At the time, Paul demonstrated that one could take a long single-stranded DNA from M13 phage, around 8,000 or so, or 7,000 bases, and fold it up in a simple thermal annealing reaction with complementary oligos to form discrete products that have the size of viruses. But he hadn't yet really explored how to apply this technology towards virus-like particles. In our lab, we publish capabilities around designing the oligos and sequences in order to fold effectively any structure on the 10 to 100 nanometer scale. That's published for DNA, and we have a paper that's under preparation for also folding messenger RNAs on similar scales. When we synthesize these, aside from the design and production of the oligos that are needed to basically hybridize or bind to that long template single-stranded DNA, we use click chemistry as well as hybridization to attach moieties to the outside or the inside that are basically complementary to barcodes, or click chemistry to directly covalently attach small molecules. The long single-stranded DNA that we're using in the applications that I'll show you, we produce bacterially using a helper plasmid system. And this is a fully scalable process using fermentation. And we also purify out endotoxins in this process to not have any unintended immunological effects. When one folds these particles, one gets pretty quantitative production of virus-like particles. This is just showing sort of a scanning microscopy image at the 100 nanometer scale of these single particles. And one can actually see from high resolution structural imaging that one gets the icosahedral viral-like shape of interest. So these are very well-defined chemical structures and moieties that offer us then the ability to present antigens and other small molecules for various applications. We recently published a click chemistry together with a label-free characterization technique to show that one could also click a variety of different compounds, including oligos, carbohydrates, mannose, dyes, peptides, PEG polymers, if of interest, as well as protein antigens for a vaccine application that I'll show you. Thinking about how stable these are, both in saline for storage, but also in the presence of nucleases. So they're stable in PBS for weeks, shown up on the upper right, but they do degrade in a matter of hours in serum. And it depends, of course, on the model of serum, but they're stable roughly up to 6 hours. The reason for this degradation is minor groove binders, DNase 1 and DNase 2, so DNase 1 outside the cell and DNase 2 inside the cell. What we've done to test this hypothesis of degradation, but also test whether we can extend the lives, is use minor groove binding compounds that basically associate and compete with those DNases. And what we see when we add this competitive small molecule, in particular diamidine, which is DAPI, you can see that in 10% mouse serum on the far right in the blue, you can extend the lifetime of these particles out to roughly a day, which for many gene therapeutic applications might actually be more than long enough. So turning to first application, that's going to be in vitro, and then I'll show you an in vivo application. As I mentioned, everyone's familiar, of course, with a very long-standing effort to develop vaccines that are effective for HIV and AIDS. And in 2010 was a pivotal moment in this timeline where two neutralizing antibodies, VRC01 and VRC02, were discovered from patients living with the disease that became really major targets both for vaccine development but also for antibody therapeutics. In our lab, we're interested in studying how virus-like particles can be used for vaccines, and there's of course a long precedence of using virus-like particles, either whole attenuated virus vaccines or protein subunit vaccines. The vast majority of vaccines that we know and love for chickenpox and measles and others, we get shots once in our lifetime, and they basically last for a lifetime. Others need to be boosted on long time scales, but these are of course the overarching goals for immunology and vaccination strategies. Part of the reason for this robust and long-living or long-lasting humoral immunity that emerges is because these viral antigens present repetitive multiple copies of antigens in highly structured ways that trigger and bind specific cognate B cells that get selected in germinal centers. And this is importantly done with intact virus that are also recycled by follicular dendritic cells, shown in the core of the germinal center reaction, where B cell clones are effectively selected and then matured over time until they get sent out to be memory cells. In the HIV area, protein subunit particles have been used to try to elicit response that would eventually produce this VRC01 class antibody of interest. That's the neutralizing antibody, broadly neutralizing antibody for HIV-1. And this is a clinical product that's still in trials out of Bill Sheaf's lab from the Scripps Research Institute. And this collaboration between Bill and our lab is also with Daryl Irvin, who's at the Ragon Institute and Koch Institute at MIT and in our department. An interesting observation for HIV-1, in contrast to this protein particle, is that HIV-1, unlike many viruses, most viruses, displays very sparse copies of its envelope protein, and in a very variable way, from just several copies out to many dozens of copies. And so it's thought that the spacing between these antigens, which aren't actually regular, but are highly variable, might be one of the challenges that we're facing with developing effective vaccines. In any case, this protein particle from Bill's lab is an engineered protein that self-assembles into sort of a 20 nanometer particle that's highly stable. And it's a germline targeting, so it targets germline B cells and is able to elicit an antibody response, which is thought would then eventually be boosted towards developing the VRC01 class antibodies. And so that's been shown in vivo as well to be robust in targeting germline B cells, but what has not been shown is really how the architecture and structure of this vaccine is either optimal or not for eliciting this B cell type response. And so there are a number of open questions scientifically that we were interested in exploring with DNA nanoparticles that allow us to test these types of questions that are listed previously, which include the copy number of the antigen on the particles, the size of the particle, the spacing between the particles, the affinity of the antigens, and so forth, in order to give us insight into what the rules might be for developing an effective B cell targeting and selection molecular vaccine. And of course, we're also interested in understanding whether these principles for HIV-1 might translate over to other pathogens as well. So I'll just highlight a few of the results from the study that's published. This is just showing B cell calcium imaging on how B cells basically in vitro are triggered by different vaccine particles. And so the first observation that we were able to make was that one sees a monotonic increase in the activation level. So that's on the right side with increasing antigen numbers. So this is 1, 2, 3, 4, 5, 10, and then the positive control, which has the 60 copies of the antigen. And so with as few as only 5 or so copies, we already see quite robust B cell activation. So that's just varying copy number, but the spacing was also changing in those particles, and so we were interested in understanding in a more simple system how the spacing between two antigens may or may not trigger a B cell in its activation. And so for this, we used a rod-like particle where this technology allows us to ask this very specific question and of how the spacing between the antigens affects B cell triggering. And we saw, again, a monotonic increase with spacing out to around 30 nanometers. Which was then maintained out to a larger distance of around 80 nanometers. So that was a surprising result because the sort of thought in the field is that you want to basically put antigens as close as possible and packed onto a particle that's presented to B cells, and that doesn't seem to be the case, at least in vitro in this system. That result of spacing also held up in a more two-dimensional or three-dimensional way in which this monotonic increase in cell activation also was elicited. Another important question is what's the effect of flexibility? In other words, the distance between the antigens and how they might change when you present. So do you want to present on a rigid scaffold or a rigid virus-like particle, or do you want to present on one where the antigens can diffuse, much like in an enveloped type layer or lipid particle? In this case, what we saw was that by fixing the spacing, we saw a significantly higher activation in this dimer in B cells than corresponding systems in which the antigens were allowed to basically change their distance. So that was another insight that's non-trivial in thinking about how to design effective vaccines, that it seems that B cells really like to see this fixed spacing between these particles. Cellular imaging also corroborated that that B cells are specifically targeted by these virus-like particles. So EOD is that red label antigen showing clustering these particles in invagination as they're endocytosed over time of 5 minutes and 30 minutes. And this is on the right showing basically compared to, again, the positive control downstream signaling of a kinase that gets activated by that endocytic process where the DNA virus-like particles similarly activate to the positive control. Now, that was all in vitro, and work is ongoing between our lab and Darrow's lab to look at how antibody titers are elicited in vivo, and also how these particles are targeted to germinal centers and which subclasses of B cells they actually get targeted to. And I think one just point to make on the way towards now an in vivo application to SARS-CoV-2 is that these particles are, of course, distinct, as I opened up the talk with, from other particles that are made purely from proteins, like the EOD GT6-temer, because we can incorporate siRNAs and messenger RNAs and ASOs directly into this modality, which is, I think, a really unique opportunity going forward, because now we can target the specific B cell subset, get endocytosed, and then put in those highly potent siRNAs of interest that Phil told us about in the last talk. So we're exploring a lot of different ideas in that direction. If anyone's interested, we'd love to brainstorm. Because as a platform technology, we're able to swap out antigens very easily to other proteins or peptides or sugars of interest. And so this is a collaboration then that started with the Lingwood and Schmidt labs, the Raygun Institute, when the pandemic hit, where instead of presenting that HIV antigen, we then swapped out the receptor binding domain of the spike protein of SARS-CoV-2. And again, use this platform of the icosahedral particle to ask questions about whether we can get B cell triggering and eventually also antibody titers in vivo, and whether those antibodies then also neutralize virus. So for this, we again did this B cell assay, but now with two different clones, the B38 and the CR3022 that some folks have heard about. And see again a monotonic increase from 1 to 6 to 3 copies compared to a very low response in the B cell assay for just the wild type monomeric protein. There's a protein nanoparticle that is still more, signaling more strongly, but nevertheless, reasonably within a similar magnitude for the B cell response. We then looked at just in hex cells expressing ACE2, the receptor of interest, that SARS-CoV-2 and this receptor binding domain also bind. We saw robust binding in vitro of the 30 copimer compared to the monomeric protein alone. Finally, then also are immunizing mice with this particle. And see, a few things is a little bit busy and has a lot on it. So on the far left is just the titers that are elicited. So there's the monomeric RBD, and then the DNA VLP with the RBD that has 30 copies on it. And this is a stabilized variant, which I mentioned was using that minor groove binding inhibitor, DAPI. And so what we see is, of course, robust titers compared to the RBD, which is promising following a boost. But we see no effect with the stabilization of these particles against these DNases that degrade them. So what does that mean? Well, it might mean that they aren't needed. And so one hypothesis is that the lymphatics are actually a highly protected environment. If you want to traffic antigens to sites of germinal centers to be able to recognize them and develop immune responses to them, it would be a bad idea to degrade things on the way. So that's one hypothesis. In any case, turning to pseudovirus neutralization assay also, these titers also in antibodies neutralize the Delta variant, which we're all very familiar with, but also interestingly appear to neutralize the more challenging Beta variant that is the one out of South Africa that's been more of a challenge. So it's early days, and as I mentioned, these are made from nucleic acids. Of course, it's early days in understanding how they elicit immune response when they get taken up. And so there are a couple canonical, well-established pathways of interest to look at that a graduate student has been studying. So the first is an endocytic uptake mechanism where TLR9 is a protein receptor that basically dimerizes and binds CpG domains, and that's very commonly used as an adjuvant to signal and elicit downstream interferons. And then another pathway that's cytosolic is through cGAS-STING, which is used in some immunotherapy applications. And so these are two of the obvious pathways that one would expect these particles to elicit when taken up by cells. So looking in vitro, just in HEC293 reporter cells, Rebecca in lab found a couple interesting things. First, she found that for cGAS-STING, when you transfect these particles, you get pretty robust signaling, but for TLR9, she did not see signaling, which was interesting. And I should mention that if you do not transfect these particles, these have no ligands on them. They're not being taken up. They basically just degrade out in blood or plasma. To look a little bit more at the TLR9 signaling, what Rebecca's done is take CpG domains that we're all commonly familiar with that are used to elicit immunological response in the TLR9 pathway, and basically present them from these virus-like particles, either 0 copy numbers or 40 of them. And when you display 40 of these oligos, you get a very robust signaling. And more importantly, even one can actually engineer this to tune the degree of TLR9 activation. So by presenting either 0 or 10 or 20 or 40 copies, she's able to selectively sort of tune up or down the TLR9 response. So the hypothesis here, we don't have a really detailed mechanistic understanding of what's going on, but what we hypothesize is that if you put just free oligo CpGs into a cell or into an endosome, you get some weak dimerization of the TLR9 protein that binds it, but the TLR9 protein actually likes to see 2 copies of these CpGs in order to robustly dimerize, and probably also cluster in order to signal. And so when you stick them on a nanoparticle where you get that right spacing and copy number again, you really are able to get this avidity effect in order to really cluster those, the same way as you get that avidity effect of clustering in B cells. So I think there's, going forward, going to be a really just interesting, sort of area of biology opening up and trying to think about how these particles can be used to elicit these types of cellular signaling responses outside the cell, inside the cell, by presenting stoichiometrically, multivalently precise ligands, whether they're sugars or proteins and peptides. Again, it's very early days for us, but we have some promising sort of in vitro and early in vivo studies showing that these particles are active in both contexts and that we have quite good engineering control of them. Future applications that we're exploring are a variety, ranging from targeting specific subsets of cells with siRNA and ASO and mRNA therapeutics, and also programmable immunostimulation for immunotherapeutic type applications. With that, I'll just thank again the collaborators who did the hard work on really the in vivo and in vitro models, and our experts respectively in those fields. And then, of course, the group members, Ike Wamhoff in particular, and Grant Knapp. So Ike, he's on the job market if anyone's hiring. It might not be the bad time to advertise. Thanks again for the opportunity to share our work.
Thank you, Mark. That was terrific. Anybody have some questions?
Great talk. I was wondering, is it possible to image the virus-like particles for the localization intracellularly, just maybe with fluorescent markers or something?
Yeah, absolutely it is. It's a great suggestion and a great idea. And so the sort of 2 lines of imaging that we're pursuing actively currently are both intracellular and extracellular. So intracellularly, using antibodies to stain endosomes as these are getting taken up and sort of see what their life is, which is more of a single cell type analysis. And one can even do super resolution, to really look at co-localization and so forth, although it's challenging. And then in Daryl's lab, we have a joint student who's also doing germinal center imaging. So basically, once these traffic to lymph nodes in the mouse, you can basically take out the entire lymph node and clear it and slice it. And look at very specifically, which Daryl's lab is really expert in, which cells are taken up or binding these particles and what their history is. So those are really important experiments to do to really understand mechanistically where these are going and how they can then be used therapeutically. So.
Great question. Any other questions?
Thank you. Thank you for the great talk. I was wondering, it seems like there's so many advantages to having these particles, obviously for human engineering efforts, but I was wondering if there's any examples of RNA or DNA particles that are encoded, that can obviously encapsulate something to be delivered that are encoded naturally. Like, has evolution ever discovered this method as a way for delivery?
Yeah, we get that question a lot, and it's a great question. So DNA is a phenomenal polymer due to its stability, for encoding information, and so another part of my lab looks at that for data storage in DNA, because fossils show us that it can last a very long time. And RNA is, of course, a much more transient molecule, relatively unstable, but similarly has this incredible information encoding capability. But I think, so I'm by far not an expert on the evolution of life, but I think proteins are really that additional layer that really protect these molecules for their value of encoding information and making sure that it gets to the right place and so forth. And so the presence of nucleases, that we mentioned. So I'm not aware, to answer your question more directly, I'm not aware of any example where these nucleic acids have been used to make virus-like structures. But before airplanes were flying, we also weren't aware of examples where steel was going to be used to make something that looked like a wing of a bird and fly us through the air. So we'll see what happens with the fate of this technology. But as an engineer, we sort of are exploring the opportunities, despite evolution's choice of selection along the way to make us as functional beings. So.
One last question.
Yeah.
Okay, here we go.
So have you tried making the virus-like particles bio-inspired, so actually designed after real viral particles?
Yeah, great question. So yeah, an undergraduate student is actually taking AAV serotypes that we know exactly what antigens are presented and what structure of the AAV. So AAV9 is a very interesting one because it transcytoses robustly to the brain, and is looking at putting those same antigenic regions onto our particles to see if we can elicit similar type transcytosis properties. If that were possible, that would be obviously an incredible capability, but it's very early days, but it's a great idea. These particles, because we design them to have known structure and chemical composition, we do, like sort of dino therapeutics that George Church spun out, have the capability to also do sort of machine learning to see what properties they have, whether it's biodistribution, safety, so forth, binding, and then kind of, by looking at libraries of these and also barcoding them, and then feed that back into sort of a system that might learn, what's working, what's not working, and kind of take a next iterative step. The same way as actually Dan Anderson, I think, is next. He's done beautiful, incredible work in this area using lipids, right? Using combinatorial lipid particles and trying to learn in those contexts as well for actual delivery to therapeutics that work. So.
Great. Thanks so much, Mark.